Dr. Jaykumar, sir?
Yeah, please tell me.
Yeah, shall I start, sir?
Yeah, please.
Yeah, very good afternoon to all. Once again, I welcome you all for the 11th day of technology-based entrepreneurship development program in aquaculture, sponsored by Department of Science and Technology, Government of India. The second session will handled by Dr. M. Jaykumar. Dr. M. Jaykumar is a senior scientist in innovation team, Skimco Private Limited, and earlier he worked as a senior scientist and head in Coastal and Marine Ecology Division. He completed his master's in marine biology from Alagappa University, Karaikudi, and doctorate in marine biology from Andhra University, Visakhapatnam. After his PhD, he worked as a scientist at Aquaculture Foundation of India for a period of 3 years. He was instrumental in planning and executing the project at root level for the fisher folk livelihood as part of seaweed cultivation in Tamil Nadu, Andhra Pradesh, and Kerala. Later, he worked as project scientist at National Center for Coastal Research in the field of ecotoxicology. After that, he worked as a senior scientist and head at C6 Energy Tuticorin, Tamil Nadu for 6 years. He also worked as marine expert and consultant for Vianant Solutions, Hyderabad for more than 4 years, and L&T Rockwell for port marine monitoring. And he also worked as marine expert and consultant in an eco-restoration project with Chennai Rivers Restoration Trust, Chennai. He is a member of Board of Studies of Department of Oceanography and Coastal Area Studies, Alagappa University, Karaikudi. He has taken part in various cruises in East and West Coast sample for marine studies. He has also worked in various environmental impact assessment studies related to port and harbors, desalination plants, etc. He has also conducted around 10 training programs on aquaculture by National Fisheries Development Board and Central Marine Fisheries Research Institutes. He has published more than 35 research papers, 1 book and 1 book chapter. He has completed 1 project on seaweed shrimp free trials as principal investigator, sponsored by Fisheries University, Tamil Nadu. With this background, once again, I welcome Dr. M. Jaykumar for delivering the topic of seaweed farming as a profitable business. Hand over to you, sir.
Yeah, thank you very much. Thank you very much for making me elaborate brief introduction. So I'm not new to this Sathyabama University. Actually, I have given so many seaweed webinar in Sathyabama. This is a part of the webinar. It's mainly for the entrepreneurship and how the development and how this seaweed will be going at present. What is the status? So I just want to say that the seaweed is it's a very good profitable business in India actually. So regarding this, I want to discuss the webinar, and I want to thank Sathyabama University for giving again a wonderful opportunity to discuss about the seaweed farming and related updates and all to share to you people. Thank you very much. So I am starting now. So now, just saying that. So is the seaweed business and seaweed farming is a profitable business? The question is like that now. So seaweed is now a wonderful and making $1 million business to everyone as a small startup to big companies. This seaweed farming is becoming a more and more viable and everyday life for everyone. So the seaweed farming, I will say the seaweed farming is a profitable venture, and now the seaweed farming can be grown up as extreme end. Last 3 decades, its development is enormous. So like if you say that there are 17 to 18 startups are there now. So for example, these are the things, this book, this 17 C6 Energy Zero Cycles left side, that has been June Entrepreneur journal, it has been published. So I'm just sharing that information to you. So there are more than 100 companies have been started now, 100 companies in seaweed in India. So in that, who was making more success, like a startups, 17 have been stated in the journal. So there are so many startups are on the way actually. So now put together, we ask ourselves, so what is the major challenge? So raw material like seaweed is a major challenge, because in a country like India, we need a more is agriculture backbone, agriculture background of crops and everything. So the biostimulant is needed. So biostimulant is the major essential for there to boost the crop actually. So instead of going for a chemical fertilizers and inorganic fertilizer, it is best to make the soil regenerate. By applying this seaweed, the soil regenerate and make them soil healthy and future generation ke liye. So this will be very useful for that time to grow up the crop. So if you take about the thing now, where we are lack actually. So we can say we couldn't be able to make a target. So how much volume we want and the constant quality and price level. So now we want India should be a leading in all the ways, all the agriculture things. So in that we have a vast coastline. Also, we can say that 8,100 kilometers of coastline it is there. Still, we are making a very minimal quantity of seaweed. Why it is happening? Because of the awareness and also only the corporate companies have been blooming over with the seaweed. So everyone should activate and everyone should do his own business, and also they can earn more seaweed in the small livelihood to the bigger sectors. So we required a large scale cultivation. So for example, making a fuel. Now actually in India, the seaweeds have been very useful for the biostimulant, sap for fertilizers. So like in a country like India, we can develop lots and lots of more product. For example, we can extract carrageenan and this phycochloride and hydrochlorides are useful for various industries application, pharmaceutical, nutraceuticals and other areas. So we require to form and we have to make them to aware of what is the seaweed business and how they will do everything. So the major cause is we require a large scale with the low cost supply of seaweed, because large scale with the low cost and require high quality and constant supply of seaweed. So these are the very important. Okay. So if you take about the India seaweed, commercial seaweed, that is ulva, kalarpa, these are the edible. Ulva, kalarpa and porifera, these are being useful for so many varieties. But kalarpa will be using for a salad, and ulva will be useful for the so many other purposes, juices and other agents. And also this brown seaweeds and red seaweeds are useful for example of biostimulant and alginate, sargassum for alginate. So I told earlier that there are 8,100 kilometers of Indian coastline. So in that 844 species of seaweeds, in that 434 red and 194 brown and 260 species are green seaweeds. So these are the total composition of India. So then we can say that they are worldwide. Worldwide, there are 10,000 seaweeds are reported. In that three major forms, that is green algae, red algae, and brown algae. In that green algae, chlorophyta, we have 1,500 species, and red algae, rhodophyta, 6,500 species, and brown algae, phenophyta, that is 8,800 species. In more than 800, in India, there are more than 800 species has been reported so far. So these are some seaweeds, as ulva, aspergopsis, and sargassum. So from how to promotion the seaweed in India. So the government initiated a Department of Fisheries, Government of India, and make a autonomous body, that is NFDB. And NFDB is giving fund to the CSI Institute like CMFRI, CSMCRI, and NIOT and state fisheries, and they will give the training, demonstration, and assist the seaweed cultivation, establishment of processing unit like that. So according to 2017 to 20, so there are 255.32 tons of wet weight had been produced, and that we can say the Kapafagis is 201.72 tons of wet weight in over of 1,000 beneficiaries to 1,500 beneficiaries, and roughly it comes around 53.55 million, that is 535.5 lakhs funding has been given to the seaweed farming. So if you take the natural components, what are the components have been there? So we can say that red algae, green algae, and brown algae, there are so many vitamins, carbohydrates, proteins, pigments, lipids are being there, and also 80 to 90% of water with after air drying is 10 to 20%, and polyphenols like flavonoids, bromiphenol, and terpenoids are there present. So vitamins like provitamins A, B, and C, and D are there already. Lipids, pigments, everything is there. And if you take about the global production of algae, so the total aquaculture, we can say that 97% are doing aquaculture, seaweed aquaculture. In this, 34.1 million tons are being harvesting in that, and remaining 3% from the wild harvest. So state-wise production in India, you can take that Tamil Nadu is around targeting 2024 to 25 as on 3 lakh tons of weight. So these are, and secondly, majorly constitute to, that is Gujarat, next to Gujarat is Maharashtra. So these are the state-based production targets in 2024 to 2025. So now we can talk about our Tamil Nadu. So how the seaweed cultivation going, what are the places it will be harvesting. And if you take that 5 districts are been cultivating the seaweeds. For example, Pudukkottai, Kanyakumari, Ramanathapuram districts, Thuthukudi and Kadalur. So if you take a major cultivation, I've been taking part in Pudukkottai and Ramanathapuram districts and part of Thuthukudi. And some areas, some patches in Kanyakumari and Kadalur are taking part. So the actively participation, for example, there are 3 different zone circular there. So our Tamil Nadu has been very lucky that we can stock or we can store the seaweeds in all the 3 districts. So this can act as a seed bank also, we can say, because if you take about the Maharashtra coast or Gujarat coast, on Maharashtra is very difficult to stock the seaweed because here we have the monsoon is a 3 to 3.5 months, the monsoon will be there. So we couldn't be able to stock the seaweed, and all the coastline is the same and same climatic condition and also the same weather. So, and if you talk about the Tamil Nadu coast, so Pudukkottai we can do culture about December to May, up to May. And if you talk about Ramanathapuram district, seaweed cultivation can do around January to October. So in Thuthukudi, we can say May to June, September till we can do the Thuthukudi cultivation. And the remaining parts of some R&D trials have been going there in the Kanyakumari and Kadalur district. So these are the rough estimation I just made according to 2022 the rate, the raft method. So actually this cultivation can be classified into 4 types. That is calm water, rough water, and medium calm, and also the shallow water. So the shallow water like a bay like Mandapam area and also in Pudukkottai, Ramanathapuram area and Pudukkottai area, we can say as a shallow water. So shallow water also the 2 different method can be done. Actually, the bamboo method is being well established and used by the Ramanad cultivators. And the pole method can be used in the shallow water in Pudukkottai district. So this shallow water itself having 2 different techniques. One can bamboo, one is pole method. So bamboo method will be doing in Ramanathapuram district and pole method will be doing in the Pudukkottai districts. So this is the cost of making 100 rafts, for example, in a kind of seed bank. So it's 2,25,000 in Mandapam. And this is a pole method I talked about earlier. The Pudukkottai district is a 7-meter model line. The distance between pole is around 2 meters. So we can, at a stretch of 150 meter, 100 meter, we can make inside the pole. And this is the total budget of the 100-meter monoline pole method. This 8,090 rupees is for the 100 monoline ropes. And this is a 100 monoline ropes at cultivation at Thuthukudi. Thuthukudi is like a plot method. So here we have to use 4 to 6 anchors and make with 8 mm rope tied with, that is a plot rope. So frame rope, that is a frame rope or a plot rope we can say. So here we are using 15-meter monoline. Same like 2-3 boys we can keep, and this harvesting period is 40 days. Because this method we can keep it in 5 to 6 meter depth. So this will be growing very good because it's open sea. Actually, it's open sea. Open sea will be a more nutrition water than shallow water. So this is the cost if you take for 100 more lines, it will be a 55,200. So it's around, if you put down 10% extra, we can put for down this cost. This is a net bag method. Net bag method is been adapting in Kanyakumari area. Our tube net method is HDB pipe method. So instead of bamboo, we are using HDB pipe. In the HDB pipe, we can use a tube net method. The tube net is around 90 dia with every 1 meter we can insert 250 gram of seaweed. So at present we will get some 10 times growth in compared to the Pudukkottai and Ramanad area. The Kanyakumari will be a high DGR seaweed. So for this 100 net bags, roughly cost estimated estimate is for 25,800. This is it's a, 2 years back. So the cost is around 35,000 it will come. So this is some of the photos of we harvested the Lipuram, Kanyakumari districts. So finally, actually we need a seed bank because we faced a problem in 2014-15. So we didn't get any seeds anywhere. So from little seaweed from 100 kg, 200 kg brought from Gujarat or some other places. So we're trying that and now the seaweed is booming now. So why this happened? Because we don't have those times, we don't have a proper seed bank arrangements. Now at present, for cultivators, because of the training and the need of importance of seaweed, now they know how to stock the seaweed in the rough waters. So why we need a seed bank? So mainly it helps to keep the seaweed live condition during the harsh weather, that we know that. And we can supply seed to known cultivators to develop the seaweed cultivation. So we can supply seaweed instead of giving 100 kg per cultivator. So you calculate 100 kg in 45 days, one cycle, it will come to 300 kg. Then 300 kg again, 45 days a cycle, again it's coming around 800 kg. So the cultivator has to wait for a period of time for the initiation of the initiation seed is very high. Then they are harvested very high after the every cycle of interval. So if you have seed bank in all the sites, it is very easy to develop the seaweed cultivation, and also the availability of the seeds and others, farmers, everything will be very comfortably, they can work over there. The seaweed is more. So the seed bank is mainly useful for the seed multiplication. So these are the 3 points will be very useful. And this is the importance of the seed bank needed. And if you talk about the land-based seaweed cultivation, so merits we can demerits also there. The demerits is the only cost expensive and one-time investment. So merits are there. So land sea-based, we had a lot of challenges to be fine because sometimes rough weather will come, sometimes epiphytes attack will be there, some fish bite and snail grazing. Then pufferfish attack, everything is there, and also a lot of natural calamities will be coming, then we lost the seaweed. So for that, we can plan to make a land-based cultivation. So this will continue. We can monitor the seaweed regularly interval and engage less manpower, and also free from the predator attacks and free from the natural calamities, and harvest seaweed at any time. And we do culture in 10 months in a year and constant yield. And we can say that to the buyer that constantly that we have some stock of seaweed and we can buy back and everything, we can do it nicely with this land-based cultivation. And this, according to that, land-based cultivation has been well established in Europe countries and other developing countries. So this is from the Australia. So the land-based aquaculture and its cultural species. There are 6 to 8 genera can be suitable in tank culture. That is Laminaria, Udendaria, Porifera, Coprophagus, Condus, Ulva, Ascophyllum, and Grassleria. So these are the species we can easily cultivable in the land scale. And this is also a land scale with the IMTA. Seaweed aquaculture for the mainly for human food and land-based human food. And this is from the Portugal. Near the shore, they make 10,000 liters tanks and they're doing cultivation. And this is also landscape cultivation in Nova Scotia, then in southwestern part. This is Acadian Seed Plant Company. They have established a well land-based cultivation. And this is the Ulva tank cultivation. So as I told, that challenge in open sea farming, that is seasonal and seed material supply, grazing and disease, epiphytism, drifting and loss of crop, climate change, inconstant yield and variability of quality of the product, no control over critical farming parameters, so farming management issue, labor issues. So these are the challenges we'll be seeing day-to-day life, day-to-day in the seaweed farming. And this is a raft method in Ramanathapuram, some photos. So see the raft sinking by overweight. If you're not harvested properly, after growing, it will go inside and it will lose the seaweed. the seaweed will break. So this is some pole method I told. In this distance between this two pole is 7 meter and this is around 2 meter. So this pole method has been well established in Pudukkottai area. This is a Thoothukudi district rough water cultivation. So this is Kanyakumari. So for example, if you take a model outline of the project, if you take for the if you go for self-help groups, so number of villages is 8 and the name of the villages you can say anything like number of people in each group will be 5 per group and 25 numbers in one village. There are 200 ropes per person and 25 cultivators per village. So initial demo training and meeting will be arranged initially. And number of trainings will be 2 training and number of cycle in 1 year is 4 cycle. So this is a small outline of the project. So 8 villages and how much they can yield and everything. So making a 200 rope per person, 25 cultivators a village. So this is economics about the 2022 as I told earlier. This 100 meter mold line is around 8,000 rupees. So dead back method is 40,000 rupees. And the raft method is 2,04,000. That is for the 100 rafts. So before going for a site selection, so we have to see some following important points, such as to identify suitable village with a favorable site selection, because some villages will far away from their site and some area sites will be good and the village will be far away. So this has to see whether going for a site, whether the site is suitable for cultivation or cultivators are there, and cultivator also we have to see the below poverty lines people will go and work. So we have to check all the details, and after that we have to say the importance. We have to make the group, and then we have to make some training and awareness, aware the people that for each districts, and we have to say that this is an alternate source of income, and you can produce more money regarding in the seaweed, like that. We have to say some important points. And also the seaweed buyback. So we have to arrange the buyback option. So many people, they will do the cultivation and they don't know how to sell and how to make a stock and who's the buyer. So we have to make the link between the buyer and the seller. And so that will be very useful, important for to making the seaweed boom in that area. So as I told, the method of cultivation, there's a calm water, shallow water, slightly rough water, and slightly rough and rough to fully rough water. So these are the method can do. The calm and shallow water is a raft already in Mandapam, Ramanathapuram area they're doing. And shallow water is a monoline culture. As there's a pole method with the monoline culture, they're doing the shallow water. And slightly rough is some Thoothukudi area. So we have a frame rough water with 15-meter monoline, and rough water is a tube net and ladder method. We'll be using with the HDB pipe with the net bank method, and also a tube net method with the ladder can use for this rough water. So these are some cultivation techniques we'll be practicing in Tamil Nadu. So these are some methods. This is a net bank method in Kanyakumari, and pole method in pole rope method in Pudukkottai, HDP pipe in Kanyakumari, and bamboo method in Ramanathapuram. So as you all know that how much the seaweed is important like that, because in general seaweed is rich in carbohydrates, which make 35 to 74% of the dry weight, like polysaccharide, several minerals like iodine, iron, zinc, calcium, lipids are present. And also protein and other amino acid pigments and vitamins, polyphenols, and other nutrients are present. So when it comes to this, I'm just taking about some little time that what are the major components are present in the different types of seaweed. So in Asia, seaweed is a part of the human diet. Actually, in China, Philippines, Vietnam, Japan, and other neighbor countries like Sri Lanka, also they're using as a food source. So only in India, we are not tried up this as a food. So now there are so many companies that are planning to make so many food products from the seaweed have been establishing now. So very soon we can also have the seaweed items in the supermarket. So now if you take about the brown algae, that is alginate is been preparing from the, has been extracting from the brown algae. So this most abundant polysaccharides, because this alginate has abundant polysaccharides and also 40% of dry weight of the brown weed. So 40% of the dry weight of the brown algae, that is an alginate, is currently using as a gelling agent in the food industries and stabilizing and thickening substrate in the beverage like cosmetics, paper industry, pharmaceutical industry, printing and textile industries. So this alginate we using in different components like beverage, cosmetics, paper, pharmaceutical and printing, and also useful for gelling agent. The most common use species from the alginate is Ascophyllum, Dudevelle, Euconia and Laminaria, Leucaena and Macrocystis and Sargassum. So here in India, we can say that Sargassum will be the more popular seaweed. So now in India, using Sargassum, their alginate extraction is going on with the snap industry in Ranipet, and also they are now the largest sargassum taking company in India now. So now with sargassum they're taking a biostimulant also. So many industries with the combination of kappa and sargassum they're making a biostimulant, and it is going well, and also this will be very making a prominent growth in the crop nowadays. Earlier, that biofertilizers are being taken from kappa. So recent years, so many R&Ds proves that better yield in sargassum. So it is better to go now. So many R&Ds are being came up, and like Aquaculture, they are making a combination of sargassum and also kappa. They're combining a product and making a good product and been selling in the market. So most of the volume of the used alginate production is harvested from the wild stock, but still we don't have a proper culture technique for the sargassum. So sargassum will be taking the wild. As of now, they are taking about 1,500, 2,000 tons roughly from the wild. And they're making this alginate used in the food industry, and alginic acid, sodium alginate, potassium alginate, and ammonia alginate, calcium alginate. These are the chemicals they've been extracting from the that is from sargassum. And also the respectively European additive codes they're giving for the European additive codes of E400 to E405. So if you see these codes, this any packet or any frames or any things, so we can automatically the product is from sargassum, and maybe combination of sodium alginate, potassium alginate, and alginic acid and calcium alginate. So these are the codes we'll be giving for the different products that we extracted from the different seaweeds. So now I'm talking about agar. So agar, as we know that it's used for gelling, strong gelling hydrocolloids, and it also used for the food additives. Generally in recognized as a GRAS by US Food FDA, and Europe is classified in the E406 additive. So each, as I told earlier, so each seaweed is having a different code. So with this code, we can say that this agar will coming under what category. So agar is a mixture of polysaccharide composed of agarose, the gelling component, and agropectin. So which have a lowest gelling capacity. The various application like pharmaceutical food, this is also having a lot of commercial and industrial and pharmaceutical use. So with 90% of the production used for the food industries, the agar. And the major constituent of from Gelidium and Grassia species. Most of the agar commercially from the commercial production from cultivated seaweed. So these are having a well-established cultivation technique, like a raft method, we can do this species. In CFM-CRI also had a trial and given training to the one-to-one cultivators with the gelidilla and grass area. So they are also having a good yield production. So, and now we're talking about the carrageenan. So carrageenan is mainly taken up from our known kappa. So if you take about the total cultivation, so kappa is been occupying more than 85 to 90% of the cultural by the farmers. So carrageenan is a hydrochloride and similar to the agar, and it's a red algae, and it represents 30 to 80% of the cell wall constitute act as a stunning agent. And it is also having a pharmaceutical uses, food uses, cosmetic sector, with the several carrageenan differing of chemical structure and the property having different use. So this is mainly carrageenan coming from the predominant countries. This is like Indonesia, Philippines. They are the two countries that have been doing more seaweed cultivation of kappa. So they're labeling carrageenan labeling as E407 as a European standard. So these are the few dishes from the seaweed. So like a sushi, sendai, and ongri, and seaweed soup, salad, potato with the seaweed. So these are some seaweed as a food they are doing. And as I told, what are the different types of agar, agro, carrageenan, and alginate. So gel forming agent and confectionery and to allow the clean slicing, canned meat product used to make a clear noodles in Japan. So these are the different types of extraction will be from the seaweed. So alginate will be dental impressive modes and immobilization application and textiles are using alginate. Carrageenan is ice cream and chocolate, thickening agent, salad dressing, air fresheners, gel, pet food, and creams and desserts. And agros are the source of molecular biology research. Agros be useful, and agar is gel forming agent. These are some useful. And apart from that, now so many R&Ds have been coming and being useful for, have been making more useful for this product. So these are some seaweed dresses. So these are sea colored dye from the seaweeds. This will be useful for the textile industry. So natural dye from the seaweed. And this is a sea salt, seaweed salt, and some alcohol drinks are also been preparing by using seaweed. Color paw is the seaweed can make alcohol. So if the predict market into 2030, so see the table that here we are seeing that nutraceutical is $3,954 million of thing it will be paid in 2030. And next to that is a biostimulant. So animal feed additives and pet food, methane additives, and bioplastics. Bioplastic also is going to come 2030 with some product of the $733 million. So this is a business prediction of 2030 and how was established everything. So this is the cultivation happening in China and Norway. So how the cultivation is going, because here the leasing policy is not there in our seas. In near neighboring country like Sri Lanka also having a leasing property. So they will lease the island and all for the cultivation. So now we have to reframe the leasing thing is there now, so everyone can go for a big farm cultivation. So these are the cultivation happening in China and Norway. So they have a seaport, like our harbor, they have a seaweed port. So seaweed port is mainly to harvest the seaweeds. These are the cultural seaweeds. It's a kelp plant. So see how the seaport look, seaweed port. So these are winches, big winches, and boats have winches, and this harbor is full of winches. So to take a seaweed out. So now, actually now for the long, the big farms and all, we don't need more labor actually. Now big farms with the mechanized farming. So many with the 4 or 5 laborers, we can farm 10 hectares, 20 hectares also we can do with the tractors. So this we get mechanized farming. So mechanized farming will be well established now in Indonesia, so and other countries, Europe, developing countries. So seaweed farming for the net bag model. This is the model of the net bag. So this is a diagram representing of seaweed farming and net bag method model. So how it looks, seeding between the two net bag is 1 meter, is the 100 by 100 meter. Totally in 100 net bags, 100 meter, 5,000 net bags we can do in 1 hectare. So this is the cost of it for seed farming net bag method. There's 1 hectare farm. So harvest period is 60 days. That is 30,68,600. And this is 1 hectare monoline farm. 10,72,412 rupees. This is the model of 100 meter monoline method of seed cultivation. So now that Pradhan Mantri PMSY schemes, they have made more allotments for the seed cultivation. So Tamil Nadu bagging of 171 lakhs. So 171 crore. Next to Tamil Nadu, we have Gujarat, then Maharashtra, then Orissa. So these are the Tamil Nadu having more than 171 crores. So now they are making a seed plant, seaweed park also established. So now as per the latest thing, seed park thing has been established, and only Tamil Nadu has been sanctioned, and still other states are being not given any reply for that. So these are some papers. Thank you very much.
Thank you, sir. Thank you for your elaborate presentation on economic importance of seed itself. Participants, do you have any questions? Participants, do you have any questions? Yes, Akshay.
Hello.
Yeah,
Yes,
Akshay.
yeah, tell me.
Yes, sir. Sir, in seaweed culture now, in Pradhan Mantri Matsya Sampada Yojana, they are promoting seaweed culture for alternative source for fishermen. But there are a lot of cases like there is conflict between trawlers and the people who cultivating this seaweed. So how to tackle this thing?
No, actually, you have to make one proposal to NFDB through your local fisheries officer, district officer, state fisheries. So before that, you should study the site and with the people who are already there and doing established, and make one small proposal. Then you can submit through state fisheries, and through the NFDB, they will get a fund alert for that. Hello?
Yeah, Akshay. Thank you, sir. So one of the participants, Dr. Stanley Abraham, sir, asked, apart from phycocolloids, which type of compound can be commercialized?
Actually, now we have carrageenan has been extracting from the kappa. Carrageenan and alginate have been extracting from brown algae alginate and carrageenan. So apart from that, we have so many enzymatic reaction is there, enzymatic extraction is there. So through enzymatic extraction, the component like which component we have to extract by, for example, if you take for kappa, so carrageenan can be extraction, then alginate like that we can extract. So still some R&D is going on. So every corporate, they are having a separate extraction, enzyme extraction technique is they're making. So apart from that, what the product they are going to mainly extract from their need, they are extracting. So actually, C6 is there, Aquaculture is there, and our company is there. So everyone they are having, we are making alginate and carrageenan. And here, if you take about the Aquaculture, they are extracting the carrageenan. So like that, everything they are having according to their view. Hello?
Yeah, thank you, sir. So one of the participants asked, kindly explain market potential in India and abroad, sir.
Sorry?
What is the seaweed market potential in India and abroad?
So here now I told no already there's so many startups are been emerging now. So there are more and more seaweed companies that have been arising now. And when compared to foreigner, already we are like 20 years, we are back now. So already in some companies, Academic Sea Plants and Sikura or some other, so many other, they need, they're doing more and more works on seaweed. Apart from that, in our India, they are listed about 25 companies are there. So 25 companies were being well established and they are doing their business. And when compared to India and abroad, so now everything same state like whatever now day to day we are getting every updates. And also if we don't have any seaweed culture here, we can import the seaweed from the Philippines and Vietnam and also in China. So like sargassum also we can import. So now the person who's having a marine biology background or a marine background, they can establish a seaweed company, and also they're doing a good business nowadays. Yeah.
Thank you, sir. So I think one more participant asked, he is working on the seaweeds. From where we can get the dry seaweed, sir? He's asking.
Which dry seaweed, which species they want?
They're not mentioned here, sir.
So dry seaweed, actually dry seaweed we can get it from some CRI you can approach. Otherwise, some companies, so is really a student means some corporate, they can help us. They can do an intern in corporate companies. So dry seaweed they will get. Otherwise, we can locally, we can engage some fishermen and we have to collect it and we have to dry it. That is the way we can do it, dry seaweed. Because as a student, R&D means you can get it here. And if your bulk quantity means we have to take it from the farmers. Farmers are selling the dry seaweeds, the bulk quality in Mandapam and Pudukota area. So we can get it from them also. Yeah.
Thank you, sir. I think there is no more questions, sir. Thank you, sir. Thanks a lot for joining the elaborate on seaweed as economical importance, sir. Thank you, sir.
Thank you. Thank you very much. Bye-bye.
So once again, good afternoon to all. The third session will be handled by Dr. T. Marudupandi. Marudupandi, sir?
Hello, sir. It is audible?
Yes, sir. Audible, sir. Yes, sir.
Thank you. Thank you,
Yeah,
sir.
thank you.
Sir. Shall I share my screen, sir?
Yeah, yes, sir. Yes, sir, please. Okay, let me introduce first short introduction of Dr. Marudupandi, sir. Dr. Marudupandi completed his PhD in Marine Biotechnology at Annamalai University, Parangipete, and Masters in Philosophy in Marine Biotechnology from Manamaniam Sundaranagar University at Tirunelveli. Dr. T. Marudupandi currently working as an aquaculture consultant at Qualitech Products and Services, Chennai. Previously worked as several state and central government institutions. He has published more than 40 research articles in peer-reviewed journals and conferences. He has a very strong knowledge and experience in the feed formulation and characterization by industry level. Moreover, he has expertise in the molecular markers for optimizing the nutritional requirement of fin and shellfishes, feed additives for improving the health status and disease resistance, hatchery technology of groupers, Litophinus vanami, Venus monodon, and marine ornamental fishes and pond culture of edible shrimps. enhance the lipid level in microalgae, nanochlorosepsis serena for aquaculture application, as well as live feed culture, microalgae, rotifer, coffeeports, and artemia. Moreover, replacement of fish meals and fish oil for renewable and natural resources and commercial production of probiotics using fermenter and microbial solutions for aquaculture application in effluent treatments. With this background, once again, I welcome Dr. T. Marudapandi for delivering the topic of economical importance of probiotics in aquaculture. Over to you, sir.
Thank you, sir. Thank you for your nice introduction. And I also thank to the head of the department, Dr. Inbagandan sir, Center for Ocean Research, as well as Director Research ma'am, as well as authority of Sathyabama Universities. So now we are going to see about the topic of economical importance of probiotics in aquaculture. So as we all know about nowadays that using probiotics in aquaculture practice has increased substantially when compared to the last times. Of course, it is maybe the awareness created with the farm in between the farmers as well as the industry has realized the potential of the aquaculture. That's why now it is increased when compared with the earlier times. It is overall probiotic values, including medicines and poultry industries. When compared to aquaculture, it is very limited. Of course, the market size in the year of 2023 is around $87 billion will be total market value of probiotics. Of course, that expected revenue for in the upcoming years, of course, 2030 would be around $220. Of course, the growth rate is remarkable. Of course, a compound annual growth rate is 14% in upcoming days during that 2023 to 2030, which denotes that it is a growth of aquaculture. Of course, in aquaculture concern, it has been classified the market as with shrimp culture and fish culture and other like pond, what do we have to call it as specific cultures, indoor tilapia, and so many culture practices are there, like indoor culture. So many things there, but mainly we are focusing in India is the shrimp culture. Of course, worldwide also shrimp culture is playing a major role in terms of economical analysis. Of course, fish also contributing substantially in that economical point of probiotic contributions. Of course, the production is very high. Of course, India has exported around 45,000 crores per year from the shrimp culture itself. So we have to imagine the potential of the market value for the probiotics. So how we can produce the probiotics? Of course, fermentation is the one of the method we can produce that probiotics commonly used in industries, because the volume will be big. What we are doing in the conic flask, it's also same, the process is same, but to increase the volume, we need some sophistication called the fermentation. All hope all would aware about that fermentation and process. That the main concept is that media, what we are going to substrate should be very less. Of course, the yield that is called as biomass final product should be very high. That is the basics behind the economic portion of the fermentation process. So before to start the fermentation process, it should be, we all have to have some basic idea about the strain selections and of course laboratory process development and pilot scale and industrial scale. Of course, a downstream process, which means called the centrifugation and product packing techniques and effluent disposal, which we are going to dealing with the probiotic modules, and other commercial consideration also has to be thoroughly studied before to start the fermentation process. Of course, we all know that isolation, source of isolation is a very important thing. So because it should be play crucial role in probiotics, because nowadays the major limiting factors in probiotics in aquaculture practice is the probiotic strains is not working in all the places, because in some places the salinity is very less. Some farming area the salinity is very high, and some farming areas the salinity is median level, like 45 to up to 5 ppt. Even freshwater also, that nowadays people are doing the farm culture. So it is the probiotic strains has to be tolerate all the kind of salinities. That is the most important factor to select the strains, so that only we are isolating the strains from different regions should be that would be working more than that normal traditional ATCC or MTCC culture collections. So of course, we all know that this is a basic steps which we are going to use for probiotic isolation. After isolation, serial dilution and spread plate method and filtration, centrifugation, these are all very common methods which we are adapted for probiotics. These are some of the sampling how we are collecting and fish gut, which means the good healthy fishes guts will be used for isolating that probiotic strains. Of course, some mangrove sediments or some good water can be used for isolating the probiotic strains. These are some specific media we can use to, as we know that millions and millions of bacteria available per ml of water. So we don't know how many bacteria is viable and how we can going to use the strains. So, but for that, isolating all the strains to we can use different medias for isolating the probiotic strains like nutrient agar medium or MRS or sobel marine, or even some Vibrio species also has reported as a beneficial effect. We can use the TCVSO for that also. These are all some common or very basic enzyme screening will be performed for a probiotic concern. Of course, we all know that any diet which is going to aquaculture practice is mainly composed of protein and lipid, and of course carbohydrate. We have to that beneficial bacteria which we are going to supply as a feed supplement has the ability to digest that feed nutrition in order to convert the nutrient as a growth through the microbial transportation. So for that, it is very basic screening method of protease activity and amylase and lipase as well as cellulase activity. These are all these four are very important. Of course, phytase also more important because nowadays soya is used as a major protein source for feed ingredients. Of course, we have to reduce the phytic acid content which is already present in the soya in order to improve the growth of animals. So these are all some toxicity assay also we have to check, because the strain which you are going to select as a probiotic, it should not be toxic to the host animal. It also should not create any environmental impact. So we have to consider all the aspects before to selecting the probiotic strains, so that we can use to here as a Artemia salinae as a model organisms. These are some strains which we have conducted the experiment for the toxicity study. Just I am presenting here for your reference only. So media for fermentation, as we discussed a little bit about it. Actually, the maximum yield should produce the media. As of course, that the production in terms of the biomass should be very high. That is the media. Has to be composed for major any probiotics or any bacteria. We have to grow by using that carbon, nitrogen, and minerals, and growth factors and precursor, which are all the very essential factor. Of course, some of you people may aware about the response surface methodology. This is actually statistical tool which we use to commonly in industrial optimization. By that, we can analyze more than 10 or 12 parameters at a time. Instead of it will be very save the time consuming, as well as we can come to arrive the exact dosage or biomass which we will get when you are using that particular media. Such a wonderful software we can use for nutrient optimization process. So these are all from a point we can reduce the production cost, because altogether ultimate any industry the production cost has to be reduced in order to it will be beneficial to both the farmer as well as the industry peoples. So normally for carbon sources, sugar cane and molasses, wheat molasses and vegetable oil, starch and cereals, glucose, so many things we can be used as a carbon source, as we all know about it. In terms of nitrogen, corn steepler is a major nitrogen source commonly used in the fermentation process. Of course, some slaughterhouse waste and urea, ammonium salts, nitrate also can be used as a nitrogen sources. In terms of growth factors, of course, vitamin and amino acids will added where the microorganism cannot synthesize them, because some as you know that these are growth factors, growth stimulating factors. Okay, apart from that, this is actually what are the components are needed for the fermentation process in the industry level. Of course, the compressed air and sterile compressed chilled and cold water, hot water, steam. Because of our sterilization process, you don't think much about it, because EB generator and equipment control money is a SCADA. We will call it a SCADA. It should be automatically flow and diagram will be there. P&ID we will call it as that will by that itself we can monitor all the process. We can operate it by using the SCADA itself. We can which valve we need a valve or bore valve. By that we can transfer the pH or whatever the solution. Instead of everything manual, we can avoid the contamination while using the SCADA type of which means automatic fermentation process. So different component is needed to do that probiotic productions. It will be very good for commercial productions. Actually, media concern actually defined media, as we know that nutrient agar composition like TCVS composition, whereas some bacteria we don't know about the nutrient requirement of the particular bacteria. Maybe it is a new bacteria. For that cases, we have to provide more number of nutrients. By that we can isolate the particular strains instead of defined media. Of course, some chemotrophs also required some amount of glucose or amino acids and vitamins, which is needed actually for as a carbon source for their growth. As well as the design media, already I told about you the designed media, complex media, which is in highly complex nutrients will be there, mechanical, TSR, nutrient broth. These all comes under the designed media. Of course, most of the pond bottom here, I want to emphasize these slides. Shrimp pond was already very polluted. Actually, after 60 DOC, so many amount of feed will be dumped in the pond. If the improper feed management will lead that adverse effect of pond environment. And of course, even if you are supplying probiotic, it won't grow in that such a pond conditions. So that bacteria, there won't be oxygen actually in the pond bottom. So that cases we will use that strains actually, which means the probiotic has to be withstand or able to grow that where the low oxygen will be there. So such a cases we will use the thioglycolate as a media components for growing that particular strains. These are some inducers. Inducers means, of course, we can use the fermenter for producing the enzymes. I already told. So along with the probiotics, some people are giving along with the enzymes. So it will be yielded more. Of course, the probiotic going to produce the enzymes. Instead of that, we directly supplying the enzyme along with the probiotic, it will be yielded a very good result. I later in upcoming slides, I will show some that farm result, which was already evaluated by China for one product that we will discuss later. These are some common inducers. Actually, major enzyme only I am presenting here, and what are the microorganisms will be used for producing that particular enzymes, which means that cellulose or pectinase protease and alpha amylase, which are the major important enzymes used in the aquaculture practice. Of course, these are the stress elements and anti-foaming agents. As we all know, that oil we can use as a top for preventing the foam formation during the agitation process. It's a more number of foam will be created, and this can be eliminated by using the sunflower or olive oil to avoid the foam formation. So of course we all know about it. Types of fermentation, the two types of fermentation can be used in that industrial solid state. Of course, any substrate will be used along with the probiotics. We are in terms of other aspects, it's a liquid state fermentation. It is a normal fermentation. In that also three types of people are employed. That's batch fermentation, batch by batch, and wet batch fermentation also there, and continuously the fermentation will be running. Whereas the continuous fermentation, we should be supply the nutrients continuously in order to maintain the cell density. So this is a simple diagrammatic representation of the fermentation process, what happened in that industrial level. So, of course, you know that seeds culture, it is a stock culture of mother culture, and then it is up to 1 liter or 5 liter or 10 liter, based on the volume of your fermenter will be used. This is at a large scale, is a 2000 KL fermenter. Then it will be broth. The fermentation end product will be a broth that can be two type of material can be used. One is a direct broth, for example, PS, photosynthetic bacteria, or CP product will be used as a liquid form only, whereas in most of the probiotic like cut probiotics will be used as a powder form. In that case, they will be used centrifugation, like a tubular centrifugation will be used commonly for centrifugation that biomass. Then it will be subjected for lyophilizing or drying, and we know that it is laboratory level, whereas here we will use as a spray dryer method for long-term storage, as well as the viability of the product in the farm conditions. So as we don't know, the probiotic, what the characteristic features is essential. It should be able to survive within the digestive tract of the host animal. That is the most important thing for that probiotic. It able to attach actually the intestinal epithelial layer has to be colonized. Of course, it should not be toxic also. It should not be toxic to the host animals. Why it is essential? Because to utilize the nutrients which is present, we are supplying through the diet. So it will be, of course, the probiotic will produce the enzymes that will convert the nutrient as a growth in terms of growth. So it is, of course, the stability of the probiotic is important. pH, how much pH it's able to tolerate it, or what temperature it is essential. Of course, storage condition, how long we can store, what is the validity we can give, all during the transportation. So what is the condition? Because in liquid form, we have to add some preservative like phosphate salines we have to use, or some glycerol based on 5 to 10%. So it is a different, it is our portion. So these are all the some major important characters which has to be considered in terms of probiotics. Of course, this is a simple thing on what are the aspects involved in the probiotics in aquaculture practice. Of course, growth is involved. We can use the probiotic for disease resistance, because it is so many literatures reported that that bacillus species or lactobacillus having the more disease resistant effects against the particular pathogens like vibriosis, as well as some papers also reported even WSS also can be controlled by using the probiotics. Of course, it is directly while supplying through the gut, it will be be improve the gut, stabilize the gut, and also it will be support the growth. Of course, that manage the pond environment. Of course, nitrogen cycle will be managed effectively by using the nitrifying bacteria. Of course, it will be greatly support to the reduce the mortality of the shrimps. And altogether, the yield should be very high. That is the most important thing. Any product, not only probiotic or disinfectants or herbal supplements, anything if you are using, ultimately for the farmer has to be get benefited. As long as farmer is survive, the industry will be survive. Otherwise, both will be got collapsed. I want to emphasize this slide here. The constraints to use the probiotics. As we all know that probiotics are beneficial effect, it will give them more growth or pond, but the limitation is there because all the strains not able to grow inside that industry. Of course, the salinity and environmental parameters are greatly influenced in the growth of particular probiotics. Of course, the part where the high pollution is there, definitely the efficiency will be reduced while using that probiotics. Of course, whatever it may be. So we have to optimize the conditions. So the strain should be selected. That's what I emphasize in the strain selection. The strain should be selected different salinity ranges. Of course, the strain has to be able to withstand the different temperature and the temperature, different salinity, and so many things as we added subsequently, and the enzyme production efficiency. Everything has to be monitored before to select the probiotics. So here I am mentioning the commonly used probiotic strains for aquaculture practice. Of course, you may all aware about the Bacillus subtilis. Bacillus species is, role is indispensable in aquaculture practice. Without the Bacillus species, you can't imagine the role of probiotics, particularly aquaculture. So such a way it will helpful in terms of growth and as well as water management and soil management played very well. Of course, Lactobacillus gut probiotic we all know about it. Even some yeast. Saccharomyces cerevisiae strains also having some potential result in terms of enhancing the growth, and menon oligosaccharides, which is present with the yeast, also having acted as a prebiotic, as you may aware about the prebiotics, which can support the growth of probiotics. So such a way it is a wonderful concept actually, probiotics, because normally while using disinfectants or some chemical drugs, definitely like why the government of India has banned the antibiotic usage in aquaculture practice, as we know that continuous usage of antibiotics will damage the intestinal microbes. Also, your normal microflora also get affected. Of course, it will create the resistant strains against the antibiotic. Then we can't manage that. Whereas in probiotic, no harmful effect will be there. It will be very environmental beneficial approach actually. So we can use the probiotics for use of aquaculture, which means altogether is a growth. See some of the probiotic products commonly used in aquaculture. I presented here. Of course, we know that Aquaclact is one of the good cut probiotics. So many companies are there, around 40, 50 companies in India. They are doing the probiotic production services. Just for a reference, only some of the probiotics I mentioned here. And of course, the role of water and soil probiotic. These are the strains, the Bacillus subtilis or Bacillus ligniformis, Bacillus megatherium. Of course, this Bacillus megatherium is very, as my experience I found, some strains, not all strains, because ATC is also supplying cultures and the MTC is also supplying cultures. Apart from all strains, that Bacillus megatherium plays a vital role in water organic load reductions. Efficiency is also very high when compared to other strains. Of course, nitrosammonia strains, we all know that the fact about the ammonia cycle, nitrogen cycle, the ammonia will be converted as a nitrite, then nitrate, and then as nitrogen gas will be to the top. So that will be, of course, here we are using some thiobacillus and the denitrificants and paracoccus denitrificants, because these are all that sulfur oxidizing bacteria in pond bottom. No, that will be very sludgy actually. After harvesting the crop, while seeing that it will be very sludge condition. So that condition, these strains will work effectively. It will oxidation process very good in that strains. Of course, paracoccus also play a vital role in that manage the pond bottom. So of course, to reduce the ammonia level is more important thing in aquaculture in terms of water maintenance. Of course, to improve the water quality, ultimately the animal will be stress free. Once it got stress, it's not at all easy. If you are over dumping of feed, also will make your animal stress to susceptible for diseases. This is some of the field results which we got while using our product. I'm not mentioning the product because it will be again will be a commercial aspect. So I'm sharing the results which we got. This is actually the bloom actually caused by that PGA, blue green algae played the role. This created some toxicity to animal. Actually, after applying the probiotic, it was substantially reduced. For these cases, that of course, the better managed practice also essential to manage the bloom. PGA is a major problem currently in the aquaculture practice everywhere. Most of the people are suffered with the PGA controlling. So phosphate solubilizing bacteria we are using here. Of course, this is this photo that ornamental aquarium. This is I given to one of my friend only. He is currently in Chennai. This is before just setting the water, and I given probiotic strains he used, and after that, we just seen that difference how it is working, because visually we have to witness it, unless otherwise not be because most of the companies are doing the repacking in this industry, and strain selection is most of the strains were procured from ATC sources only. So that only we have to, that's what I told you, that strain selection is the most important part in aquaculture system, because if you are producing more amount or more volume of probiotics, because the strain don't have the ability, so altogether all the process will be waste. So the strain selection is the most important and prime criteria for isolating the probiotics. So these are some basic results which we got from our products, which we evaluated in our R&D end. So for disease resistance, we discussed about it. So some bacillus actually bacillus is opportunistic pathogens. Even Vibrio also opportunistic pathogen. I am frankly telling you, Vibrio is not at all a serious disease as long as you are maintaining your water quality and other parameters very pakha. Because when you are the over organic load is there, that manipulation will be there very high. That will be increased. So management of the practice also consider as a major things in terms of disease resistant probiotic selection. So these are some strains which are already published and reported only. I am claimed here. These are some potential strains because some Bacillus cereus are reported, even it is working against that and WSSV, but mode of action, the author has not explained the mode of action of the serious process, because of course it will be produce some viable enzymes. By that it will be killing that enhance the innate immune system. As you all know that shrimp has the innate immune system. By that other like lysosome activity, SOD activity, superoxide NBT activity will be increased substantially when using the probiotic that we have evaluated. Such a way it will be working to control that any bacterial diseases or viral diseases. Of course, will be these strains are mostly used for the disease cases. Lactosine or bacteriocin. So most of the bacteria producing bacteriocins. That bacteriocins also showed very good antibacterial activity against the particular strains. Some hemolytic ability also we can observe while using that hemolytic agar. These are all again I came to that resume to the topic economically. So how the farmers are getting benefited. Really, they are benefited while using the probiotics. This is the most important thing, because already farmers are as you some of you people may aware about the production cost of the pond preparation. Of course, 10 to 15 lakhs will be incurred towards preparing the 1 acre pond per crop. I am telling one per crop, which means 120 days. Sometimes it will be 90 days. Based on the farmer, it will be decided. Based on the count only, they are going to harvest it. This is the product which already evaluated in the pond trial. 4 pond for that they have used 2 pond they used as a control, and another 2 pond they used as a experiment, which means that Aquastar pond and Aquastar pond sign. I already told you that the probiotics along with the enzymes, both they have given, and the stocking density here is around 50 shrimp per meter square with the weight of 1.4 gram animals, which means around 3.37 lakhs shrimps they have stocked in their pond. The trial actually carried out for 57 days with that weekly I had told. No? So next slide, I will show you how much dosage is required. Normally people are using. So 1 kg they have used for weekly applications though. So this is actually result of what they have got for the when compared to the control ponds and experimental pond. See this after harvesting. After harvesting, the soil condition will be very good while using the pond bottom along with the enzymes, which means that the probiotic is working effectively against the soil condition. Whereas the control pond, the P slide no, there is the control ponds that was clay very slide and sulfur formation is there. So it is showed that the probiotic can effectively manage the pond bottom. So if you are managing the pond bottom, definitely you will get the very good successful crop, because most of the problem, of course, initial stage, no problem will occur normally. Anyone can like do the shrimp farming up to 60 DOC, 60 DOC, 60 day of culture. Whereas after 60 DOC, a lot of problem will be created in the pond environment. It is not like a indoor culture what we are doing in the RAS culture or bioflak methodology, because everything in our control. Whereas in pond culture, everything is it depends on environmental conditions, because algal as we know that after 60 days feed will be increased substantially, the animal growth will also increase, subsequently feed will be increased. So over feeding will be there, the nitrogen source will create the over bloom. So after that bloom created, no, 1 week every time it will be settled in the bottom, after settled in the algal crash. So that animal most of the time will not prefer to take the even pellet feed, because it will natural, no, animal behavior is natural feed. So it will ready to take that crashed algae. The crashed algae again will be accumulated in the hepatopancreas the animal, then it will produce some toxicity, and by that it will animal may attain even 40 to 60 days itself, sometimes got white fecal, or sometimes it will be load or poor growth and elevation of ammonia. So the problem will be very huge. Actually, farmer after 60 to 90 days or 120 days, they will be spending their most of the mm, energy for securing that culture. Actually, that is a very crucial period of the culture. So, but while using the probiotic, they can avoid maximum or around 80 to 90% of the problem, because it is an environmental way, effective, ecologically way approaches. Whereas while using disinfectant, of course, it will cross the algae. Some places people will use to for PKC, that benzyl conium chloride as a chemical is there for managing the pond conditions. So the pond will be managed, whereas subsequently the animal will also growth and activity will be suppressed by that chemical, because as we know that all chemicals have some toxic effects. So like viranil, so many disinfectants are available, but of course it all are not advisable while doing that shrimp farming. Whereas the pond preparation time, you can use it no problem, any disinfectant or chlorination or copper sulfate, whatever you can add in your pond, nothing harmful going to happen for animal, because after adding that aeration, after saturation only we will introduce that animal in the pond. Whereas after 60 DOC, we should not add any disinfectants or like a chemical disinfectant to the farm conditions. Of course, it will create the most stress to the animals. So that is very most important factor which we have to consider. So this is actually result what where they are got while using that Aquastar product. Actually, it is approximately 83 gram per animal they have got while using that probiotic processes. Of course, speed conversion, which means FCR, is very less while using the probiotic, which means 0.91 only. That is a very good result actually. This is some one product, because all product almost similar results only we are getting. Actually, what is the provided dosage and applications are required for per shrimp culture per acre? Only I am. This is a model. Actually, you won't get this data anywhere in India or anywhere in the place, because this is actual probiotic application practice commonly adopted in India. Okay, pond depression time commonly once water and soil probiotic people will be used at the cost of around average value only. I'm putting here, because it's more than 3,000 also probiotic product available in the market. Of course, less than 1,000 also available. So this is an average value only I am presenting here. The pond preparation for a soil and water probiotic 500 gram will be required. At the pond preparation time, they will be spent around 3,000 rupees, and weekly dosage for probiotics. Weekly water probiotic, for example, this is I'm 120 days I am mentioning here, because people will be harvested even 90 days. I'm once again told you tell you that 20 days or 90 days based on the count of the animal, because price will be fluctuation will be there. So farmer will not be stand with a single approaches here. Of course, that weekly soil probiotics will cost around 12,000. Water probiotic will comes around 2,000. Cut probiotic weekly dosages, two dosage will be used actually, 3 gram. So that only I mentioned here 250 gram. So altogether, the for 33,500 rupees they will be spent for per crop. Sir, whether they will get that ROI, as a return of investment, they are spending 33,500 rupees for their crab, whether it is really beneficial to them. That's the most important thing. Actually, this is the Indian farming area. I am mentioning here that total shrimp farming area in India around 1.60,000 hectares are doing currently farming officially, but unofficially it will be around 2 lakh hectares, which means in the acres I am mentioning. Overall cost, if you accounted, this is overall business value of Indian culture, probiotic current practices, because everything will be varied based on the practice also. For fish culture, it is different. This is only I'm presenting here shrimp culture. Okay, this is around 1,300 crores will be involved in that sector, particularly on shrimp culture. So this is a model based on the previous result. This which is actually exact model I'm presenting here. Actually, per acre pond, the stock intensity of 2 lakhs plus bonus, which means 2 lakh seed shrimp seeds plus bonus means for they will give around 50,000 rupees. 50,000 numbers means, because of course mortality will be there, so that only I am counted as here 2 lakh as here final harvested material. The animal weight will be 25 gram, which means 40 count. So 40 count price is 400. I mentioned here. Of course, it will be now it is very less, whereas it may be increased or decreased, plus or minus 50 rupees. So just a model I want to present here, because total material we can harvest from per acre of 5 ton material we can harvest it. I already mentioned in the previous year, probiotic production cost is around probiotic cost they are spending farmers by around 33,500 rupees. So growth attained by the probiotic 3 gram per animal, which means the animal itself around 600 kg growth they will be gained because of using the probiotics. So which means it is around, you just calculate that in the terms of biomass of 600 kg into 400 rupees, around 2.4 lakhs rupees farmer will get benefited while using probiotic. Of course, I reduced actually FCR also will substantially decreased, which means that normal FCR is 1.2 to 1.5, whereas while using probiotic we can manage with 1 up to 0.9 to 1. Most of the reports are available. So based on 1, if you are reduced, you can save, or 1 means 1 ton, we can save here 5 ton. Actually 1 per 1.2 we record around 6 ton paid. So 1 means you can reduce the 1,000 tons. So because per kg is 100 rupees, so 1,000 means of course 1 lakh we can save by using the probiotics. Of course, profit per crop using the probiotic itself around, you will reduce the time and only account as a probiotic. Then we have to minus that 33,500 rupees. So the profit itself around 3 lakhs 65,000 rupees. The farmer definitely will get as a net profit because of the probiotic alone. So such a potential is there. Now only farmers are understanding. Of course, some demerits also. I say no. All the places the probiotic strains are not working effectively. That is the major limitation of probiotic. But the industry got now awareness. Instead replacing of chemicals, and of course some phytochemicals even also they are ready to replace with the probiotics. So farmers think that while using the probiotic, definitely they will get very good profit in terms of our their culture practices. Of course, for this model will be a very reliable model actually, which we prepared from the pond itself, based on the pond result only. This actually what is the cost will be incurred for producing the biomass in fermenter. So actually, this is 2,000 liter fermenter. We are putting that cost, how much it will be incurred, carbon, stipler, or lepton, glucose, the media composition of the products and all. So total biomass we can get only 25.7 kg. Of course, normally per kg will be based on the trend CFU. We will call it as a CFU in the biomass. Based on the biomass, we can give up that 500 kg, which we have seen in previous slides of dosage. So 500 gram. For 500 gram, we can give a manufacturer cost maximum. Will everything packing and stickering, transportation, overall cost will be, as a manufacturer we know that maximum 70 rupees we can finalize, or 100 rupees. So whereas it is sold out in the market up to 2,500 rupees, because the shrimp culture industry is having different business model, because B2B will be there some places, B2C is also there. So based on the business module which they're going to adapt at the particular company, they will decide the price. Actually, anyhow, any probiotics maximum 500 gram will be that 100 to 200 rupees the maximum fine. Even they are using 10 strains or multi-strain combinations or whatever it may be. But as a manufacturer, you will get very good profit. Of course, farmers also going having benefit more than that farmer. More than that company people. So what they are getting benefit out of it. So both peoples will get benefited in terms of manufacturers as well as farmers. Both are essential like a two-eye, so that we can save the shrimp culture industry or like aquaculture industry by using the probiotic in an effective manner. Effective means economically profitable. Everyone is doing culture for getting the money in terms of profit only. So definitely it will be support while using the probiotics for aquaculture practice. So thanks for the opportunity which is provided by the Satyabama Institute of Science and Technology, and of course authorities and DST Department of Science and Technology for funding this scheme. And thanks once again for all of you. Thank you. Any question, please throw it, please, sir.
Thank you, Dr. Murugapandi, sir, for your elaborate presentation on probiotics as economical importance in aquaculture sectors. Thanks a lot, sir.
Thank you, sir. Thank you, sir.
Any questions from the audience sides? So actually, one question to ask from the audience: what is the optimum value of CFU per ml for any probiotics?
Any probably at least it should be about 2 billion CFU per gram. Sir.
Okay, thank you, sir. So I have one questions actually.
Please, sir, please.
So in generally for aquaculture, so EHP is recently is an emerging disease, right?
Yes, sir. Yes, sir.
Is there any role involved in probiotics? So,
yes, sir.
Is there any control of probiotics by using EHP infection?
Yes, sir. Actually, probiotics EHP is reported by Vibrio parahemolyticus toxicity.
Okay.
Of course, it will be damaged to the hepatopancreasis actually mainly.
Yes.
EHP also like a heart for shrimps, hepatopancreasis.
Okay.
Once the hepatopancreas got damaged, subsequently it will be entered that into the intestinal microvilli also subsequently.
Definitely, microbiota also changed, right?
Yeah, it changed, and the villi also will greatly affected intestinal microvilli. That is the nutrient transportation. Whatever your feed you are giving, so slow growth is the major problem, sir. AHP.
Okay.
Growth won't come because that the nutrients what you are taking your animal no feed, that will not be digested, which will be digested. That can't be able to absorb by that intestinal villi because that intestine will be also damaged by that AHP toxicity.
Okay.
So that we have to spare two mode of action we have to take on advantage. Very barely probiotic alone will not support that whole process recovery. So subsequently we have to give the medicines to improve that intestinal villi, microvilli of the shrimps.
So we have to add the antibiotics as well.
No antibiotic. No, some herbals are there.
Herbals,
antibiotics are strictly banned in the by CAA in the aquaculture industry.
Okay.
So some herbal molecules are there which has the capacity to rejuvenate the intestinal villi. Also has the potential to manage the hepatocytes. Also relieve the toxicity from the hepatocytes. So such products are available, but not at launched as much as confidently because the virulence is varied. As you know that bacteria, some places where good water quality condition is there, that virulence will be very less, whereas poor water quality, which means pond water is very poor. That cases that activity, even 50 colony numbers of Vibrio para also will be a serious pathogen. It will kill all the animals. Okay. Whereas in good condition, water condition, even 500 colony is there. That time also no mortality will happen. Only that plaque melanization will be there, here and there. But that is the thing. That's what I told you. That organic matter play a vital role. In the management of AH or anything. So if better management practice itself, we can reduce it. If you want to cure it, we can. That also we can. It is possible.
Thank you. Thanks a lot, sir. Thanks a lot.
Thank you, sir.
So one more questions from the audience sides. Is it possible to mix water, soil, and gut probiotics?
It is possible. Anyhow, it will going to give the positive result. Of course, it is unnecessary. Why for gut probiotic for water because nitrosamine is not required inside the animal body. Again, it will be collapsed the intestine because where and how. That's what they are categorized as water probiotic. That's what they categorized as a soil probiotic and gut probiotics. So gut probiotic animal should be only supplement through feed as a gut purpose. not for water management. Of course, it will do, because anything, however, it will going to release the enzyme only. That enzyme will reduce the organic matter which is present in the water column.
Okay.
So it will be beneficial effect, because farmer side it is an unnecessary cost of production will be added. They are not ready to spend. So specific mode of action will be more beneficial. In terms of beneficial aspect, I am telling, we can use it scientifically, no problem, nothing going to be harm to the animal. But of course, the specific approaches will be give more yielded than that common practice.
Okay. Okay. So some of the questions asked from the audience side in the chat box. I'll read it out. One questions: liquid probiotic or powder probiotic? Which one suggestible for gut pH control?
For gut, we have to be used powder form, because liquid form can be used for only water conditioner or soil conditioner, because we will use phosphate supersaline. Anyhow, your bacteria, like I already mentioned, lactic acid lactobacillus species, it is actually producing acid only after entering the lactic acid bacteria. That is, name itself is there, because pH will be reduced, because low pH only required where animal intestine for the better enzyme production.
Okay. Thank you, sir. And liquid probiotic, no, sorry, any liquid probiotic increase DO level?
DO level concern, no, sir. No liquid probiotic available. So as of now, as my knowledge, because any probiotic if you apply it, definitely it will be consume the oxygen normally.
Okay.
Because unwanted some anaerobic bacteria is there, they don't consume. Actually, that's what I said. Paracoccus or some diabacillus or some bacteria are there. Strictly anaerobic bacteria. That no need to use that aeration, or that don't have any relation with the aeration. Whereas normal bacillus or some aerobic, some facultative anaerobic or facultative aerobic will play the role, not improving the DO, whereas it will consume the DO.
Okay. Okay.
Because for their metabolic activity.
Okay.
So DO level will be decreased while using the probiotic. So that only we are normally recommended the probiotic application in the morning time, not evening time, because not evening time, even phytoplankton present in the water also will consume that DO which is present in the water. Of course, because no sunlight will be there for that photosynthesis. So definitely it will depends on that complete water column. Of course, that greatly DO will be reduced, while so that probiotic application also in the morning time will be more good.
Okay. Thank you, sir. Participants, do you have any more questions?
Sir, is there any equipment to measure the dissolved oxygen level in 24/7?
Actually, some sensors are available, a DO sensor, but as you know, that Winkler's method, titration method is only currently employed in that pond level. Some titration kit is there. You can't, or else we have to develop the sensors, electrolyte based sensors. We have to based on the chemical reactions. So we can install actually. Based on that, Norwegian technology adopted one company. So you may have heard of Oxygenox, one company is there. They are doing that aerator inbuilt with the aeration sensor, which means a DO sensor. The DO sensor inbuilt with the aerator. Whenever your pond getting low DO, that machine will be automatically switch on. Then whenever it is over saturation, means above 6, it will be switched off. So efficiently it will use that aeration, because that you know very well the power cost is a major portion adding in that shrimp culture. So is there, it is available in the market.
Thank you, sir. Participants, any more questions? Okay, once again, thank you, Dr. Mardupandi, sir.
Thank you, sir. Thank you, sir. Thank you for the opportunity, sir. Thank you all.
Yeah, thank you, sir. Participants, tomorrow we can move on to the 5th module, business plan preparations. Tomorrow we have the 2 talks. The morning session will be handled by Mr. Naveen Krishna, the topic of preparation of business model conveys, and afternoon session will be handled by Dr. Govindaraju, sir, in the topic of startup and aquaculture. So meet you all in the tomorrow. Okay, thank you.
ஜி. Thank you. 2:30 டைம் எல்லாம் நல்லா இருந்துச்சு ஜி. சரியா சரி. ஆஃப்லைன்ல வருவா போதும் ஜேபி. லாலையோ ஓடுற ஓட்ட ஒலிம்பிக்ல ஓடுற மாதிரி இருக்கு. நல்லா இருந்துச்சு ஐயா. நல்லா இருந்தது. சரி சரி சரி. வீட்டுல இருந்து போறேன். வீட்டுக்கு வந்துடுறேன். இன்பா சார் இருக்காங்களா லைன்ல? ஓகே.